welcome to the Science Speaks Conversations on Health podcast. This podcast series has been developed by the Basel Research Centre for Child Health in Switzerland to share some of the great work that is taking place and to look at what it means to be a scientist within the various different disciplines that are represented by the centre. In this episode, we are delighted to be talking to Lester Frei and Dr Beichen Gao. Lester is currently a PhD student in Professor Sai Reddy's lab for Systems and Synthetic Immunology at ETH Basel. Beichen was previously a PhD student in this same lab and now works as a data scientist at a spin-off company from this lab called EngImmune. Lester and Beichen, thank you very much for joining us.
Oh, thanks for having us.
Yeah, thanks for having us.
So in this podcast, we're going to be discussing protein engineering, a technique that is central to the work that both Beichen and Lester carry out in the development of antibody therapies for a range of diseases. Beichen, perhaps you could start by explaining what protein engineering is and what it achieves.
Yeah, a good starting place to think about proteins is they're like little Lego blocks that's everywhere in nature that has specific functions that are crucial to life. So, and what we know about proteins is they're built from these 20 amino acids and things in nature. And with these amino acids, how they're built, it really dictates their final shape, their function. And actually, the goal of protein engineering is taking what we know about the shapes and functions of proteins and really pushing that to really create new proteins that might have different functions or actually improve certain proteins to sort of work better. So, for example, usually a lot of the things we look at is binding. So proteins, if you think about it like a certain pocket, again, with the Lego piece idea, you can make a square pocket that could basically accept rectangle, square, some other shapes. But what we can do then is to go back and, either by changing the amino acids, really turn that pocket into maybe a circle, and then that'll accept something else. And so that way we've engineered a new protein that can work in another biological process.
So when you talk about protein engineering and the kind of reshaping of the proteins to perform different functions, you mentioned amino acids. Mm-hmm. What is it that you change about the kind of configuration of the amino acids in order to carry out protein engineering?
Yeah, so actually, that's a good question. What we normally aim to do is by changing in different amino acids. So let's say it's always based on a certain sequence, right? So we can have A, B, C, D, which is what's seen in nature. But instead, maybe we can create a different function by making it A, B, E, F as sort of two new amino acids. And by doing that, we can actually create these changes in the final protein to do what we want.
Okay, great. So how was this technique developed? Kind of what is the history of this? When was it invented? And what's happened since that first inception of the idea?
Yeah, Lester, you can...
Yeah. So protein engineering really started, I would say, about around 35 years ago. And so what we haven't touched on with proteins is that they are encoded by the DNA of a cell. So what this means is that a cell, like all the cells in our body, they will just read their own DNA and then produce the corresponding proteins based on this. And this is important because it's not possible to directly sequence proteins. So what we do in protein engineering is we rely on sequencing the DNA and thereby then reconstructing the protein sequence based on this information. And so really how it started out, protein engineering, is people came up with very clever ways on how to link the DNA sequence and the protein sequence. And based on this idea, people have developed several different ways of doing this. And this is just like in general, it's like called like different display platforms. So when we talk later on about display platforms, what it really means is it's just a way to link the DNA information and the protein information together. And yeah, over the past like 35 years, obviously there has been a ton of innovation in this field. Also like very crucially in 2018, the Nobel Prize in Chemistry was partly awarded to like a display platform. And to highlight like how this works and what we actually mean by like the different display platforms, let me just give you like an example with like yeast display, what we mostly work on in the lab. So first of all, like for yeast display, or I guess for any display platform, we construct a library. And what we mean by library, it's just a collection of different DNA pieces which encode for like different proteins. So like what Beichen mentioned before with like the simple example of like you have one protein that's like ABCD, and then a second protein is ABEF. And just like the sum of all those different variants is what makes the library. And so we then, for yeast display, we put this library into the cell, and then we just use the natural like machinery of the yeast cell to produce the protein variants. And then from this pool, we fish out variants with like novel and improved functions. So I guess back to Beichen's example, like if we want to have like a new variant that instead of like binds a square pocket, binds a round pocket, we can isolate this from this pool. Mm-hmm. And then ultimately we just get out the DNA, sequence it, and then we know which protein does the job.
What was the very first application of this technique? And can you also talk about kind of where we've gone from there, some of the other applications that this technique is used for, to kind of really provide some context?
So one of the early applications, and that's still used today, is to find novel therapeutics. So like one really big therapeutic that was developed early on using display is an antibody called Humira, and it's used to treat rheumatoid arthritis. And like this is like a blockbuster drug. Like in 2021, there was 1 million prescriptions. And it was so like groundbreaking for like treating rheumatoid arthritis that was in part the reason why the Nobel Prize was awarded for this technique. And so that was like back in the early 90s when that happened. Still like today, a lot of display platforms are used to find like novel antibody drugs.
Yeah, I mean, we see these in not only antibodies, but for example, I mean, even other industries like food also actually use a lot of protein engineering that people might not have expected, right? Synthesizing new, well, this sometimes sounds a bit bad, but synthesizing new chemicals to use in our food. But, you know, of course, for, you know, things that are perhaps a bit more natural and potentially less harmful and more effective. So, yeah, there's a lot of times where protein engineering is used, but we don't really, you know, see it or are aware of it.
Know that it's there. Yeah. Can we go back to antibody engineering? Can you just kind of briefly provide an explanation of what an antibody is and what it does?
So antibodies are a very crucial part of our immune system. Like just naturally, they already float in our body. And they're these proteins that are like really geared towards binding targets. So for example, if you have like, say you have a flu infection, then your body will recognize this and it will produce antibodies that effectively bind this virus and then thereby neutralize it. So it's like naturally used by the body to defend itself from external agents. And what people then did is basically in antibody engineering, we just take this like framework of an antibody and we just re-engineer it so it binds like novel targets that like are not already naturally bound by the body. So
you talked a little bit about using protein engineering for like food. You also talked about it a little bit for antibody engineering, particularly in relation to treating rheumatoid arthritis. Are there any other kind of uses that kind of spring to mind that you could talk about to give us a bit more of an idea about the way in which this technique has had implications for people's lives?
Yeah, I think for me, one of the things that often come to mind is sort of new antibiotics, because this is also one of the sort of pressing issues that we have in the world. the need to discover sort of novel antibiotics that can counter what we're seeing with antibiotic resistance in a lot of diseases that we're having right now. So again, a lot of these novel antibiotics themselves are just little proteins. So they, again, have different shapes, and their goal is whether if you wanted it to enter the bacterial cells and sort of destroy a particular mechanism that's really crucial for their survival and growth, we're engineering that. There's also some that will also specifically target certain bacteria. So, right, one of the things you have when you take these heavy, we call them broad spectrum antibiotics, is, you take it, they go into the gut, they basically kill everything that's in there. So you have sort of all of these side effects that are quite unpleasant. But, the goal of engineering these new antibiotics is they can go in and only kill the bad ones that you don't want, and sort of keep the good ones there alive and thriving. And again, that requires this engineering for that, specificity there.
And yeah, another everyday topic would be allergy to cats. So the way people who are allergic to cats is because cats secrete a immunogenic factor in their saliva. And so when they clean themselves, what happens is they spread it to the fur. And so what people have done is they have engineered antibodies that target those factors that elicit the allergic response. And thereby, again, with the shape, they made an antibody that specifically recognizes and binds the shape. And so what people did is just they mix it into the cat food. Mm-hmm. And so ultimately it neutralizes this allergic component, and thereby making the cats less allergic to people.
That's amazing.
I think it's a blockbuster cat food.
So we've talked about lots of different uses for protein engineering, and I think we're going to focus more in on antibody engineering for the remainder of our discussion. Because I believe that is a technique that you use in your lab and in your research. Can you provide a bit more of an explanation of what that technique actually involves? What is the method? What is it that you actually do in your lab when you're engineering proteins for the purposes of antibody production?
Yeah, so in our lab, we've got quite a few projects always ongoing that's around immunoengineering, as we call it, or synthetic immunology. I think one of the good ones that comes to mind is, for example, we had a previous PhD student in the lab as well, Dr. Derek Mason, if he's listening, hello. So there, what he took is a really popular antibody that's used in breast cancer called Herceptin, and basically wanted to explore, okay, what can we do with, protein engineering platforms, for example, like yeast display that Lester introduced, to sort of find, based on Herceptin, to make it better. And basically how we would do that is, all right, so again, we're talking about the libraries. We can start with Herceptin. We can make these sort of really big libraries where we have, mutations across this sort of recognition part in the antibody, right? So we start to build, again, like the square pockets, the triangle pockets, the circle pockets, something. And, sometimes these libraries are huge. They can be in the millions, the billions, if not more, right? And having these libraries in hand, what we can do is then we go and we call them, select them, right? So one of the things we wanted to do was to have really improved specificity. So Herceptin targets HER2. It's a different receptor that's really a major target in breast cancer, right? So we actually just combine our library that we have with the HER2 protein, and again, we can fish out what's really, really specific, and we leave those unspecific ones behind, and then we can improve it. Sort of next steps of looking for maybe those that are stable under heat, certain other conditions. We can then also test that with that library. So we're coming from trillions, maybe we get down into 100 million, and then from that we get down into the thousands, and sort of we could engineer it that way experimentally.
What does that process of kind of getting down the number of different possibilities like involve? How do you do that?
Yeah, I mean, there's different ways of doing this, but I guess that the easiest way to think about this is we call it sorting. Mm-hmm. It's really like you interrogate every single variant on their ability to, like in this case, like with Herceptin, to bind the cancer component, and thereby it's a very effective way to like separate the two populations of like the ones that do bind versus the ones that do not bind.
So is it an experimental process or a kind of computational process?
So for, I guess this stage is like all purely experimental. But yeah, I mean, then in the next step, it's really where the computational part comes in. So when we have those like the variants that still do bind, then that's really the point of where like the machine learning into AI comes in. Because what Beichen mentioned before, it's like you have those like enormous libraries, and like really these days, like the limitation in the whole like protein engineering field is that you can only look at so many individual variants. It's like you are limited, even though the whole sorting process, it is very efficient, but there is just like an upper limit to what you can like feasibly do experimentally. And yeah, that's really then the point where you take the information you gathered from the experimental part and you take it and you feed it into the machine learning models.
Yeah, machine learning is kind of this broad term that's getting thrown around in, I mean, every discipline where we're using machine learning. But basically what it comes down to is we're creating these algorithms. Some of them are really complex, some of them are quite simple. But the goal is, right, I've got 10 different objects, 10 different sequences that we have for proteins. 5 of them are good, 5 of them are bad, right? We're going to challenge the machine learning algorithm to say, all right, can you figure out what are sort of these patterns that's in these sequences that might be more associated with sort of, in our case, binding than the non-binding proteins.
So you're basically kind of feeding, correct me if I'm wrong, you're feeding your machine learning algorithms with some data. Mm-hmm. They use that data to learn, and therefore they can then predict which of your kind of combinations or structures of proteins are going to perform the function that you want them to perform in the best possible way. Does that kind of summarize it?
Yeah. It kind of loops back to what I mentioned earlier about like the limitations of how many individual variants you can look at experimentally. It kind of bridges that gap because the machine learning can then predict like millions and billions and trillions of variants that you just cannot, it's not feasible to actually experimentally screen. So you kind of, you try to overcome this limitation.
So you were both part of a team of researchers that received funding from the BRCCH back in 2020 to carry out research in response to the COVID-19 pandemic. And I bring this up because I know that this project that you're involved in used protein engineering in a kind of an attempt to respond to this new virus. Can you explain what the kind of the rationale or the need for that research project was? You know, what is it specifically about the COVID-19 pandemic that we didn't know, the problem that, you know, we had that required you to carry out that research?
I mean, I feel like there's nothing necessarily like unique about like the COVID-19 pandemic. It's like it has a lot of like the virus has a lot of other characteristics like with other viruses, like it mutates super fast. I guess what's really novel about it is that it's like such a global that it was such a global phenomena. And yeah, so I guess like one big problem was that, you know, the virus mutates super fast. And especially also the thing is like if you have like a new antibody therapy that like you want to use to protect like the populations at risk, What happens is if you use this antibody, it will just favor the virus to mutate around it. So then you just like evade this antibody, and which made it so that like all the antibodies that were approved really like super quickly again lost their efficacy.
Yeah, it was kind of like playing that what's it called, the Red Queen hypothesis, or the
basically that you always have to run to stay at the same place.
Yeah.
It's like you have to like continuously like put in effort just to remain at the same place, otherwise you would fall behind.
Yeah. So with COVID-19 at the start, we didn't have any immunity against it, and that's why it was so bad. But then as we developed an immune response, the virus would then start to mutate even faster to escape our immune response, right? And like Lester said, we started to see all of these rapid mutations start to emerge. It was also the first time that we started to really have the tools and the amount of data to sort of, we call it surveillance, right? To watch it mutate throughout the months, throughout the years, and throughout all the different, countries and the main question there was, okay, as it's continuing to evolve and escape our immune response, then what can we do? If we're looking for antibody-based therapies, how can we make sure that we're finding ones that won't be escaped immediately, some ones that won't, lose efficacy in 6 months' time, which then we need to find a new one and start to develop it again and produce it and get it out into the public. And then by that time, maybe it's mutated again, and that's also rendered useless. So yeah, it's kind of playing that game.
And that's where your project came in,
yeah.
I guess.
Yeah.
So how did your project kind of respond to that need? What was it doing that was, unique or different, or, you were given these capabilities for the first time in a pandemic because of the capabilities you had around protein engineering? How were you able to tackle that problem?
I think a good way to describe it actually is we've basically flipped what we normally did on its head. So normally what we would always do is we have a single target, like again, this HER2 for Herceptin, and we sort of make a library of antibodies and try to figure out what's going to bind better against that single target. But instead here, we're basically taking a set of antibodies that have shown clinical efficacy. At the time, some that were basically a bit more experimental. And then we made a library of sort of the SARS-CoV-2, the COVID-19 virus's spike protein on its own. So not the live virus or anything, but just that spike protein and a specific part of it that's targeted by antibodies.
So yeah, we just took like the main component of the virus and we just put it on yeast. And then we went from there. We used the method that I explained earlier with like yeast display.
Mm-hmm. And from that, right, we could get a subset of variants that escape antibody A. We have another set of variants with mutations that potentially escapes antibody B, C, D, right? So after a while, we get this sort of data set of, okay, which mutations are potentially going to cause escape against maybe 1, 2, or a bunch of different antibodies. Right, and this, I think, was kind of the new thing that we did, because at the time there were other groups that had made sort of, again, yeast display libraries of sort of the spike protein, but what they've reported mainly are just the effects of single mutations. All right, so let's say if we have a single amino acid that's changed, and they did find some crucial ones that really, like, with only a single mutation, one of the early antibodies would basically completely lose its efficacy. There was like over 100-time reduction in its sort of neutralization, in its protective abilities. But the question was, all right, as we start to get more and more mutations, what's going to happen, right? Because it's not always going to be a single mutation that causes escape, but perhaps a combination of 3 that really causes a change in its shape that then causes a lot of escape. right? And what we could do with our data set was to potentially see that, which I think was really cool.
So we've talked quite a lot about the amazing research that you did. And by the sounds of it, it was very impactful, made a huge difference to the COVID response. But we haven't really looked at all about kind of who was involved in that. Obviously, you both were, but who else is part of the team of researchers that carried out that research? Who else contributed to it?
I mean, these were pretty big projects. We had, so for the latest publication on the computational part, we had some of the other PhDs in the lab, Jami. We had a lot of master students who have come by and helped us out at little steps along the way. And on the experimental side, I mean, mostly Lester led a large part of it. But we also had a postdoc, Dr. Eddie Irvine, who came in and helped out.
And what about your backgrounds and your route to where you are now, Lester? Where did you start off, and kind of what was your trajectory in order to end up as a PhD student in Sai Reddy's lab?
I mean, so for me, I guess like the trajectory really started back in high school. So I was really interested in natural sciences in general. Early on, I was thinking about going on to study chemistry. But then when I was like learning about molecular biology, I was really amazed by it. Like I found it like really interesting to learn about the molecular components that make make up life. And so, for me then, so after high school, I went to ETH to study biology first. And then later on, I did my master's in biotechnology here in the department in Basel. And so then I ended up doing my master's thesis in the lab, and just stayed on after that.
So it's a good place to be.
Yeah.
And what about you, Beichen? What's your story to where you got to now?
For me, my background is also more wet lab. So, I went to uni back in Canada, London, Ontario. It's the second best London in the world. And there I studied physiology and pharmacology because I was really curious about drugs and how they worked in our bodies. And then went on to do a master's in molecular biology. And then started working actually in a company where we were sort of developing and finding antibody therapies. And there I was using these display techniques. So not yeast display, it was a different thing called phage display, which we won't get into, but it's similar, same, but different. And kind of there, I basically saw, in the process of doing the experiments, we would still sequence the variants, the antibodies that we would select at the end, but I felt like we were just throwing data away. And I was really curious about what we could do if we just took everything, right? Every step in the selection, we took the good, the bad, and maybe everything else in between. What if we could train these sort of machine learning models, and how much better it could be to help our discovery efforts. So then kind of from there, reached out to a few labs, ended up finding myself in Sai Reddy's lab and kind of working on a few projects there that started off experimentally, but then, at the very end, I was pretty much just at the computer doing all the machine learning models and all the computational components, which is still good. That's exactly what I wanted.
And in the kind of the time between when you finished your master's and you were working and you came to Sai Reddy's lab, kind of how did the space change? Because I think machine learning has grown as a discipline, as its capabilities have got markedly bigger. How have you seen that development? What's the scale of that?
Oh God, I mean, the way that I think about it, I think for you probably the same, Lester, is in protein engineering. If we go back to that, it's almost like pre-AlphaFold and post-AlphaFold. So for anyone who's listening who might have not heard about this, basically one of the biggest challenges in protein engineering is, we have all of these sequences. But one of the things that we really need is to know how these proteins look like, right? Because there is still that underlying structure that we could see, but it was really difficult to do because you had to sort of purify, you had to crystallize these proteins, you had to put them under these like crazy microscopes. And it was always a challenge. You could really never predict protein structures. But then back in 2020,
late 2020.
Yeah, late 2020, AlphaFold came onto the scene, and basically it was this model that was trained on all protein sequences that we've ever sequenced and seen, not only humans, from all species, plants, bacteria, animals, everything. And what we found was it was probably the most accurate predictor of protein structure. And it was, I think at the time, there were these competitions where most models would perform at like 40, 50% accuracy, if anything. And this one was like 85% accurate. Yeah, I guess that's kind of the exploding point in the field. I mean, before that, it was still kind of exploratory. A lot of publications would still use it, right? It was really useful for a lot of engineering projects, but AlphaFold was the turning point.
And given how much it's grown in that time, Lester, what do you think the next big thing is? Where can you see it going? This is, I know, quite a difficult question to ask.
I mean, there is a few like really big fields. I feel like one of the big fields, I think, will be some sort of like library on library screening. So like what we described here is with like yeast and like how pretty much all or almost all display platforms work is you have like, say, your yeast, and you have your protein that you have in yeast. And then you use like your breast cancer target, your autoimmune target, and then you use that to find like novel proteins that like bind to it. But it's always this like many versus few problem. So like in yeast, you have like tens of millions of variants, and then like you have like one breast cancer target. Mm-hmm. And so like one like field of like interest and active research is, could we have like systems where you have like two libraries? So like, say one is your antibody library and the other is your COVID library. And then you would do like an all by all screening at the same time. So instead of having to do like massive efforts to like only one target at a time, you could test like, I don't know, millions.
Wow.
Yeah.
So it's true to say that the field continues to evolve and develop.
Yeah, definitely.
Yes. Yeah. Not to mention, I think the other thing that immediately came to mind, again on the experimental side, is actually protein sequencing. So one of the restrictions is we have to sequence everything, genetically, so with the DNA. But one of sort of the new things that's currently being investigated is, can we do the same but with the 20 amino acids? So instead of the 4 bases that we see in DNA, we've got the 20 amino acids, and is there a way that we can directly feed in the protein and just have it read out what this would be? Because that would also solve a few bottleneck issues that we have in the field, and would sort of give us a lot more data to work with.
What difference do you think these developments are going to make for society at large?
I would say like there will be a lot of like you'll have like, it probably will fuel like drug discovery. So like having more and better therapeutics, also against like really difficult targets. But also like, like even like outside of, I guess, what we in our work focus on, there's a lot of other fields where where it will have like a huge impact, like for biofuel production, for greener and more sustainable processes, especially like industrial process, where having like being able to like substitute like really toxic chemicals with like novel proteins will also be like, will just make the whole process more sustainable.
Yeah, and it'll speed things up massively, I think. Right, we're seeing, I mean, about a decade ago, we were still sequencing sort of out of the libraries that we had constructed and evaluating clones individually, but now we're scaling up to be able to test out thousands, and then plus ML, we can test out even more, right? And I can only see it sort of improving from here, getting much, much faster. So in terms of engineering all of these new proteins.
I feel like in 50 years, people will look back at what we do now, and they will wonder like how did we get anything done? Like how was it possible to get anything, like any new like antibodies and any data? So yeah.
What do you mean you had to do all of these rounds of sorting, and
it's like
it
the way
took
we
you 6 weeks to get?
It's the way we look back now at like the 1970s and wonder like how is that possible? Like how could they get anything done? But it worked.
Yeah.
But without that, we probably wouldn't be where we are now.
Yeah.
Beichen and Lester, thank you both very, very much for taking the time to talk with me. I really enjoyed our conversation. It's been a real pleasure discussing your research today.
Thank you.
Thank you.
Thanks for having us.
Thank you for listening to the Science Speaks Conversations on Health podcast. This podcast was produced by the Basel Research Centre for Child Health in Switzerland and hosted by me, Vicky Edkins. Editing was carried out by Sebastian Schell at the University of Basel's New Media Centre. Thank you to our generous funder, Fondation Botnar, and our partner institutions, ETH Zurich and the University of Basel, for your ongoing support. If you would like to learn more about what's happening at the BRCCH, you can visit our website at brc.ch for information on our upcoming events and to sign up for our newsletter. We're also on social media. Follow us on LinkedIn at the Basel Research Centre for Child Health, or on X, formerly Twitter, at BRC_CH.